Hello, hello.
Hello.
Oh, hello, hello. Yeah, good morning. Okay, so today we will continue with the thermophysics. Okay, yep. So we just have a few questions remaining. Okay, yep. Yeah, here. Okay, so there are three states of matter: solid, liquid, and gas. Explain how the temperature of a liquid changes when it boils and it solidifies. Okay, yeah, so this one should be quite straightforward. Okay, remain constant, remain constant. Yeah, okay. So we can consider the heating curve. So when we increase the temperature, okay, and then we have melting, and then we increase the temperature again, and then we have boiling. Okay, so the temperature is always constant. Okay, during the change of state. Okay. Okay, state how the separation of the particles in liquid changes when it boils and solidifies. Okay. Okay, let me show the picture. Okay. Yep. Okay. So we are very familiar with that. Okay. So when it is boiling, okay, okay, when it's boiling, we are going from liquid to gas. So the separation is going to be increasing. Yeah, okay. The separation of the particles will be increasing. Okay. Solidifies. So going from liquid to solid. So in this case, the separation will be decreasing. The particles are getting closer and closer. Okay. Next, explain what happened to a liquid during evaporation. Okay. Yeah. Okay. So we mentioned about this yesterday. Okay. Evaporation. Okay. This one. Okay. Okay. So what happened during evaporation? So there are particles with more energy. So the more energy, they have enough energy to escape. Okay. So this is the main point. Okay. More energetic particles, okay, will escape from the liquid surface. Okay. So they will come out. So this leads to the average kinetic energy of the liquid falling. Okay. Because of the more energetic molecules they escape, so the remaining molecules they're going to have less energy. Okay, so if they have less energy, the temperature of the liquid will decrease. Okay, so the overall temperature will decrease. Okay, so that's why evaporation has a cooling effect. Okay, so it cools down the rest of the liquid. Next. Air is a poor conductor of thermal energy, but a good insulator. Give three everyday example of use of trapped air to provide insulation. Okay, so we are familiar with that. Um, yes. Okay, so trapped air, for example, the double glazing of the glass. Okay, so a layer of air will reduce the heat loss. Okay, across the window. Okay, or we can put the insulation in the wall. Okay, or we can put the insulation in the roof and so on. Okay, okay, example, double glazed window, what we mentioned just now, cavity walls. Okay, um, so we have cavity in between the wall, and then we can fill in with polystyrene and things like that. Okay, fur or fleece jacket. So in our jacket, we have fur or fleece, which can also trap air and keep us warm. Okay. String vest, fiberglass for roof insulation. Okay. Yep. Okay. So string vest maybe not very familiar. So let me just find a picture. String vest. Okay. Uh, okay. Yep, yep, yep. Okay, hold on. Okay, okay. Uh, okay. Yeah, so it kind of look like this, okay? String vest. So it can trap air in between, so it can also have the effect of keeping warm. Okay. Next. Using trap air for insulation is more effective than using air alone. Okay. So what's the reason? Okay, because transfer of thermal energy by convection is restricted. So because the air cannot move, so the heat cannot transfer. So we can keep warm because of that. Okay, describe an experiment to measure specific heat capacity of aluminum block. Okay, so let's say we are given a metal block or aluminum block. How can we work out the heat capacity? Okay, so we did discuss that. Yeah, here. Okay, so measuring heat capacity of a solid like a metal block, okay, or a liquid, okay. So the method is slightly different. Okay, so our main objective is to find, okay, so to calculate the heat capacity, we need energy, we need mass, and then we need temperature change, okay. So from the experiment, we can get these three information which will allow us to calculate the heat capacity. Okay. Yeah. So how we do the experiment? Okay. So first we have the setup which look like this. Okay. So first insert immersion heater power P into the center of a cylinder of metal and place a thermometer into another hole in the cylinder. Okay. So one hole we put the heater, another one we put the thermometer. Okay. Switch on the heater for a measured time. So we know energy equals to power multiplied by time. So we know the power of the heater and then we know how much time we turn it on for. We can calculate the energy. Okay. So energy divided by mass and the change in the temperature. Okay. So work out the energy supply will be P multiply T. Okay. Record the temperature of the metal before the heater is switched on and the highest temperature reached by the metal just after the heater is switched on. Okay, so we can have the starting temperature and then we can have the final temperature. Okay, so we can say this is initial temperature, final temperature. Okay, so now the change in the temperature will be final temperature minus the initial temperature. Okay, so we get that as well. Okay, work out the temperature rise of the metal, which is delta T or delta theta. Measure the mass of the metal. Okay, so we can use a weighing balance to work out the mass. Okay. Yep. So now we can substitute the PT. C equals to PT over M delta temperature. Okay. Yes, like this. Okay, so we can calculate the heat capacity. Okay, error in value obtained is due to loss of heat to the environment. Okay, so the value that we get, sometimes it will be different to the actual value because we have some heat loss to the environment. Yeah, okay. Next. Okay, this question is about conduction. Use the kinetic particle explain model to explain how thermal energy is transferred through a solid object. Okay, so we did discuss that. Okay, yep. So we know that we have two methods of the uh, heat conduction. Okay, yep. Okay, these two. Okay, let me copy it. Um, yep, two method. Okay, and this one. Yep. Okay. Okay. First one. Okay. Atom in hot regions pass on their vigorous vibration. Example. So the heating starts here. So this one it will vibrate vigorously and then colliding with the neighbors. So now the neighbors they also start vibrating vigorously and then colliding with the neighbors. So now more particles are vibrating faster. So this is how it spread. Okay. Neighboring atoms in colder regions. Okay. And then this one happens in all material. Okay. All material can happen for this. Okay. And then for metals, they have free electrons. Yeah. Okay. So metals have free electrons that move faster and further in the hot region. Okay. So they can move much faster. Okay. And then can transfer heat energy uh, that energy to atoms in a cooler region. Okay, very quickly. Okay, so it can happen very quickly. That's why metal is a good conductor of heat. Okay, explain why poor electrical conductors they are also poor thermal conductors. Okay, yeah. So poor electrical conductor they are also poor thermal conductor. So they always go together. Okay, so we know that that is because both electrical conductivity and thermal conductivity, they depend on electrons. Yeah, okay. So poor electrical conductor and poor thermal conductor, they do not have free electrons available to move rapidly through the material, transferring energy. Okay, so that's why they are always they always come together, poor electrical conductor and poor thermal conductor, because they do not have the free electrons. Okay, so now with this, we finish all. Okay, we finish all the workbook questions for Unit 2 of Physics. Okay, cool. So this is completely done. Okay, yeah. So this is completely done. So yes, so thermophysics is done for textbook and workbook. Okay, so now we will move on to biology. Okay. Okay, so next topic in our plan, transport in plants. Okay, so this is a very straightforward topic. So I think we can finish quite quickly. Okay, transport in plants. Okay, so how material is moved around in plants. Okay, so plants need a lot of things like water, mineral, sugar, amino acids. All of these things they need to transport. Okay, they need a good way to transport the material. Okay, so we have two main types of transport. Okay, transpiration and translocation. Transpiration is mainly about the movement of water. Okay, so movement of water from um, the root to the leaf, and then the water is lost by um, transpiration. Translocation, movement of sucrose and amino acids in the phloem from sources to sink. Okay. Uh, okay. All right. So we have two types of tissue that are responsible for transport. Okay. So xylem will carry water and mineral ions. Okay. And then another function providing support. Okay. Phloem carries food substances, sugars, and amino acids. Okay. So yes. So structure of xylem and phloem. Okay. So we need to know for the roots. Okay. Cross section of the stem and then cross section of the leaf. Okay. So these three situations we need to know where, okay, where it is located. Okay. So for the root, xylem is at the center, which form a star shape with phloem around it. Okay, so phloem is outside. Okay, so at the stem, the tissues will be oval shaped. Okay, oval shaped vascular bundle with xylem inside and phloem outside. Okay, xylem inside, phloem outside. Okay, so just need to remember that. Okay, so in the leaf, we also have vascular bundle. Normally, xylem above the phloem. Okay, so xylem at the top, phloem at the bottom. Okay, of the vascular bundle. Okay, so this part is straightforward. Okay, next, structure of the xylem vessel. Okay, so basically, xylem they are both tubes. Okay, so they are both tubes um in the plant. Okay, xylem vessel. This is what it looks like. The tubes. Okay, so they are long, thin. Okay, so one section, one section, one section like that. Okay, long and thin arranged end to end to form tubes. Okay, when they mature, the cell will be lacking cross wall. Okay, so the cross wall will be removed. Okay, so they remove the cross wall, so they make a long, continuous tube. Okay, so no problem. Makes it easier for the water to pass through. Lacking cell contents, so there no cytoplasm, no nucleus. Okay, so and then the wall become lignified. Okay, so lignified means they are um strengthened. Okay, strengthened by this lignin, which is quite hard. Okay, so this is quite useful because it will keep the tube open. Okay, so the tube does not collapse. Does not collapse easily. Okay. Yeah, so the main function, transport of water and mineral ions, okay, from the roots to the leaf. Okay, so the lignin will provide strength for the stem and then makes the vessels waterproof. Okay, yeah. Next, water uptake. Okay, so how um the transport of water happen? Okay, so it starts at the root. So the root hair cell, they are tiny, tube-like outgrowth form on young roots. So they are very numerous. So there's lots of them to increase surface area of the root for absorption of water and mineral. Okay, so we know what the root hair cell look like. Okay, so it looks like this. Okay, next. Okay, so this is one experiment that we can do. So we can put example celery in the colored water. Yeah, like dye colored water. And then after that, after we done that, we will see this, okay, xylem vessel stained with the blue dye. Okay, so it shows us um, the location of the xylem. Okay, so how the water is transported. Okay, next. Okay, passage of water through root, stem, and leaf. Okay, so how the water um move from the root. Okay, yep. Okay, so first water passes the root hair cell to the root cortex cell by osmosis, then reaching the xylem vessel in the center. Okay, so basically it starts um the water starts in the outside. Okay, and then it moves through the cortex, and then it goes into the xylem. Okay, so that is step one. Okay, so it goes into the root. Okay, next it reaches the xylem, travels up this vessel to the stem and leaf. Mature xylem cells, they have no cell content, so they are like open-ended tubes. Okay, yeah, so we can look at this picture here. It's clearer. Okay, yes. Okay, so basically we start at the root. Okay, so root will take water from the soil. Okay, so water goes into the root, and then it goes into the stem, which have the xylem. Okay, water is drawn up the stem to the leaf. Okay, so water go up. Okay, and then it reach to the leaf. Okay, so when it reach to the leaf, okay, in the xylem, then it goes to the stoma. Okay, and then it goes out. Okay, so this is in the end, water evaporate from the leaf. Okay, so this is how water is transported. Okay, okay, so there is a free movement of water in the xylem. In the leaf, water pass out of the xylem vessels to the surrounding cells. Okay, mineral ions also transported through the xylem. Okay, next, transpiration. Okay, so transpiration is the loss of water vapor from leaf. Okay, so the water will evaporate from the leaf. This is what we call transpiration. Okay, yes. Okay, water forms a thin layer on the surface of the mesophyll cells and evaporate into air spaces in the spongy mesophyll. Okay, so we can see a bit clearer. Okay, so this is another picture. Okay, which help us see clearly. Okay, what is going on. Uh, okay, yep. So first, okay, water will enter the root from the soil by osmosis. So water will enter, and then now it goes into the xylem. Okay, goes into the xylem by osmosis, and then it goes up. Okay, so a point that we will come to a bit later. So this is what we call cohesion. Okay, so cohesion means attraction between water molecules. Okay, so there is attraction between water molecules, which forms a continuous water column from the roots to the leaf. Okay, so the water molecules, they are pulling one another. So they pull together, so they go up together. Okay, in the stem. Okay, so now it will pull the column in the xylem of the shoot and the root upwards. Okay, so it keeps going until it reach to the leaf. Okay, so what happened at the leaf? Um, okay, so now the water in the winds of the leaf upward and outwards. Okay, so now the water reach to the um vascular bundle in the leaf. Okay, tension pull water from the winds into the apoplas surrounding the mesophyll cells. Okay, So now the water goes to the mesophyll cell on the surface, okay, and then the water evaporate, okay. Once it goes to the surface, it forms a thin layer and then it will evaporate to the outside, okay, and then finally it goes out, okay. So during transpiration, water vapor diffuse out of the leaf through the pores called stomata, okay. So this is the detailed process of the transpiration, okay. So we can take a look at the notes, okay, how it happened. Okay, yep. So there will be high concentration of water molecules inside the leaf, so water will diffuse out of the stomata, okay. So here we say that it's diffusion at the leaf. It is diffusion because there is no cell membrane, okay. When there is no cell membrane, we call it diffusion. When there is cell membrane, it is osmosis, okay. Water evaporating from the leaf causes suction, pulling water up the stem, okay. So what's happening here, okay, so when water is going out, okay, so when water is going out, now it will start to pull more water to here, okay. So it basically it helped to pull the water from the root all the way up the stem and then to the outside, okay. Yeah, so because we keep losing water here, so it will pull water from the rest of the plant, okay. Next. Okay, so the loss of water in transpiration is related to the large surface area provided by the mesophyll cells. Okay, so we know in the mesophyll cells, okay, here, okay, so we can see there's lots of air spaces in between the mesophyll cells, okay, which help with the transpiration and then large surface area, okay, created by the mesophyll cells for evaporation to happen. Okay. Next. Yeah. Okay. Interconnecting air spaces in the spongy mesophyll and the stomata. Okay. So the interconnecting air spaces. Okay. Next. Water movement in the xylem. Okay. So it's talking about how the water move. Okay. Water evaporating from the leaf create a suction, which what we call transpiration pool. Okay. So now water is leaving here. Okay, so now here it starts to be the water is getting less. Okay, so water getting less, then it will start to pull from the rest of the plant. Okay, so that's what we call transpiration pool. Okay, and then water molecules are held together by force of attraction is called cohesion. Water forms a column drawn into the leaf from the xylem. Okay? Yep. So just now we mentioned the water molecules, they have attraction um, between the water molecules. So the water column, it does not break because we are going against the gravity. So if there's nothing to pull the water up, the water will go down. Okay? So we have this what we call cohesion. Okay? Attraction between the water molecules that helps to pull the water up against the gravity. Okay. Yes. So this create a transpiration stream pulling water up from the root. Root xylem vessels, they are tiny tubes drawing water up the stem by capillary action. Okay. So this is what we call capillary action. So the roots will produce a root pressure forcing water up xylem vessels. Okay. Because it at here, okay, we are constantly losing water at the leaf. Okay, so here we will have a low pressure of water. So here we will have a high pressure of water, root pressure, which help to push the water up. Okay, so all of these factors help the transpiration to happen. Okay. Yes. Okay, as mentioned before, water does not travel through xylem vessels by osmosis. Does not. Okay, because there are no cell membranes in the xylem cells. Okay. Yep. So no membranes. Okay, next. So why this transpiration is very important? Because it helps to supply all the cells with water. Okay, so all the cells need to be supplied with water. Okay, so when the cell they have enough water, they will be turgid. Okay, so this is what we call turgid. They get enough water. They have a continuous water supply. Okay, so if the amount of water lost from the leaf is greater than the amount taken into the root, there will be water shortage. Okay. So when they have water shortage, it will be like this or like this. Okay. Flaccid. Okay. So the plant is losing water. Okay. So the cells will become flaccid because they are lack of water. So they will lose their rigidity, no longer press against each other. So when the cells they are not turgid, they cannot press against each other, so they will become soft. Okay, so they cannot stand upright, so they will wilt. Okay, so they will wilt. So this is not a good situation for the plant. Okay, so which means the plants need a constant water supply. Okay. Next sample question. Okay, describe how the structure of xylem is adapted to its function. Okay, so what are the special structure of the xylem that make it um very suitable, okay, for transport of water. Okay, so we know xylem look like this. Yes, okay. So this is the structure of the xylem. Okay, okay. So structure of the xylem. The cells join together to make a long tubular structure, and then there are no cross walls and no living contents. So the water and mineral salts can pass through freely. Okay? Yeah. Next, describe the mechanism of water movement through the xylem. Okay, so how water is moved. Water moved by the pull from the leaves caused by transpiration. Transpiration pull. Xylem vessels, they are very thin, so they act like a capillary tube, helping to draw water upwards. Okay, like what we mentioned just now. Okay, next, rate of transpiration. Okay, so how fast the transpiration can happen, it depends on the temperature and wind speed. Okay. Okay, so let's say we increase the temperature, the particles, the water particles, they can move faster. So we have increased transpiration rate. And then if we have a wind, okay, nearby, so what happened? The wind will blow away the water vapor. So now there is lower water concentration here, so transpiration can happen more. Okay. So in general, when we have a higher wind speed, okay, increased wind speed, the transpiration will also increase. Okay, next. Yes. The factors that result in change of transpiration, it relies on diffusion. Okay, so when there is example here just now, lower water concentration here, the transpiration will increase. Okay. Okay, so some experiments that we can do to investigating the effect of temperature and wind speed. Okay, so apparatus used for transpiration investigation will be called potometer. We have two types, weight potometer and bubble potometer. Okay, so it looks like this. Okay, so this is a weight potentiometer. So example, we can have two plants with let's say 10 degrees Celsius and 20 degrees Celsius, 30 degrees Celsius, something like that. We will set up the three plants and then we will check the weight loss. Okay, we'll check the weight loss maybe over 2 hours, something like that. So, um, if the weight loss is more, means there is more transpiration. Okay, so example, maybe 30 degrees, there will be more weight loss. Okay, because water leaving. Okay, so we expect 30 degrees to have more transpiration. The weight loss will be more. Okay, 10 degrees Celsius will be less. Okay, yep. So this weight potentiometer involve measuring the weight loss. Okay, and then we will wrap the plant with the plastic bag here. Okay, because we only want water loss from the plant. Okay, so we don't want water loss from here. No, we want water loss from the plant only. Okay, so that's the idea. Okay, next. So this one is what we call bubble potentiometer. Okay, so how it works. So at the start of the experiment, we have the air bubble here. Okay, so after some time, the evaporation, the transpiration happen. So the water is lost. So we expect the bubble will move up. Yeah, okay. To here, for example. Yeah, okay. So now we can investigate how fast the bubble move. Okay. So that tells us about how fast the transpiration. Okay. So example, for higher temperature, we expect the bubble will move faster. Okay, because the transpiration is faster. Okay, so bubble potentiometer, we check the rate of uptake of water by the movement of the bubble recorded over time. Okay, so let's say we can check the bubble, how fast it move in 2 hours, something like that. Okay, next. Ah, yes. Okay, so now we will look at the effect of temperature, effect of humidity, effect of the wind velocity. Okay. Okay, let me just bring a picture. Okay, this one. Okay. Okay. Yeah. So how we understand what is going on. Okay. So this is the stoma here. Okay, so this is the stoma here. So the water can go out, okay, in this direction. So here we can check the water concentration here and water concentration here. Okay, so this is inside and outside. Okay, so how much water? Okay, so now increase in temperature, increase kinetic energy of the water molecules. So they diffuse faster. The transpiration will be faster on a hot day. Okay, so if the temperature is high, so the water molecules they have more energy, so they will move faster. Okay, going out. So transpiration increase with increasing temperature. Okay, so we can see here when the temperature increase, okay, the transpiration will increase. Okay, but at some point it will be constant because okay, we have limited number of stoma. Okay, so each plant, each leaf, let's say they have like 50 stoma. Okay, so because it's limited by how many stoma, even if we increase the temperature more, the the water molecule they cannot move faster because they are limited by this stoma. Okay. Next, increase humidity, lower the transpiration rate. Okay. So increase humidity. So let's say outside we have more water than inside. So the water, they cannot come out by diffusion. Okay, so that's what we call humidity. So high humidity means the water amount is high on the outside. Okay, so the water cannot move out easily. Okay, so increase concentration of water molecule outside, reduce the concentration gradient for diffusion. Okay, so yeah, okay, increase, yeah, so we can see here, um, when we have low humidity, okay, here this is low, this is high, okay, so when we have low humidity, outside, okay, is dry, okay, so outside is very dry, so water will go out faster, okay, but as we increase the humidity, then outside is getting wet, okay, more wet. So if outside is getting more wet, more water, so there is a lesser tendency for the transpiration to happen. So it will decrease until in the end there is more water on the outside. So transpiration cannot happen. Water concentration is greater outside in the leaf. Okay. Yeah. So it will decrease. Okay. Next, wind velocity. Increase in wind speed will lower the concentration of water molecules outside the leaf, increasing the concentration gradient diffusion, so the transpiration rate increase. Okay, so what happened here? Okay, okay, so here when we have okay, hold on. Okay, yes, yes. Okay, so let me explain one by one. Okay, so regarding the temperature, okay, so temperature, okay, temperature outside is high, so the water molecules, they have more energy. Okay, so more energy, so they can uh diffuse faster. Okay, so this one, temperature increase, transpiration also increase. Okay, so that's the explanation. Oops. Okay, hold on. Okay, so next we explain about the humidity. Okay, so first we have the low humidity. Humidity. So outside is dry, inside is wet. So the water will come out. So the transpiration is high. Okay. But as we increase the water, so here water more and more. So now inside and outside will be almost similar, the water. So the transpiration will be decreasing now. And then when we reach here, outside there is high humidity. So outside have more water than inside. Now the transpiration cannot happen anymore. Okay. So that is about humidity. Okay. The other one. Okay. Next. Okay. So finally, we talk about the wind velocity. Okay. So you see the diffusion, okay, the transpiration, once the water come out, it will kind of stay here, okay? When it stay here, it gets more humidity, okay? So the transpiration will slow down. But if we increase the wind speed, okay, increase the wind speed, the water will be blown away. So water is blown away. So here it becomes dry again. So now transpiration can happen. So that's the reason why when we increase the wind velocity, the transpiration will happen because it will blow away the wet air. So we are left with the dry air here. So when this is dry, transpiration can happen. Okay. Next. Yeah. So this one is experiment we can do regarding transpiration. Okay, so we have four different conditions. Okay. Um we have four different conditions here. Okay. So now both side covered with petroleum jelly. So this one, for A, both side covered with petroleum jelly. So both side. Okay. So means all stomata, they are covered up. Okay. So they are covered up. So water cannot go out. Okay. So they close the stomata with the petroleum jelly. So transpiration cannot happen. Okay. So this one, we expect the least transpiration. Okay. Upper epidermis covered. So B is only up. Okay. So this one, both side covered. Okay. So this one only up covered. C is lower epidermis. Okay. So the lower epidermis covered. Okay. This one, both not covered. Okay. So we expect this one will be the highest transpiration. Okay. Yep. So this is the type of experiment that we can do. Okay. And then for this type of experiment, there are some variables that we can control. Factor to control. Okay. So means we need to stay constant. Okay. Okay. So the for the experiment to be uh fair. Okay. So we need to have a fair comparison. Okay. So means we only change one thing. Okay. Which part of the leaf is covered? Other things it need to stay constant. Okay. So example, this one we use a large leaf. This one we use small leaf. Cannot. Okay. So the result will be not accurate. So everything we need to keep the same except this part. Okay, so example, the size of the leaf it need to be same because the different size leaf will be different surface area. The type of leaf must be like, let's say we use rose. Okay, the rose plant, the leaf, all of them must be rose plant. Okay, because the different species will be different number of stomata and different thickness of cuticle. The time, okay, so let's say we leave it for 30 minutes, all of them will be 30 minutes. Okay, because the longer they are left out there, the more water will be lost. Okay, so amount of petroleum jelly smeared on the surface must be the same for each surface. Okay, so let's say we put 0.5 gram for each surface area, so all of them will be 0.5 gram of the petroleum jelly. Okay, because the more jelly we put, the more protection from water loss. Okay. Next, temperature. So we need to keep all the leaf in the same temperature because the transpiration depend on the temperature. Air current. So we know the wind speed also can affect the transpiration. So we make sure they are in the same environment. Transpiration dependent on wind speed. Okay. So we need to be very careful to control, to keep constant these variables so that we have a fair comparison of the experiment. Okay. Okay, next we look at translocation. Okay, so how translocation happen? Okay, so just now we look at transpiration, so now we look at translocation. Okay, so translocation is the movement of sugar and amino acids in the phloem. Okay, from the source to the sink. Okay, so what it means by from source to sink. Okay, source is where material is released. Okay, where it is produced. So normally it is the leaf. Okay, because the leaf will produce the sugar from the photosynthesis. Okay, so the sink is where they are transported to. Okay, so maybe the leaf will produce the sugar, and then the sugar will go to the roots. Okay, for storage purpose, or they go to the uh growing parts. Okay, the growing parts, they will need more energy, like new leaf, fruits, and seeds. Okay, or used for respiration or growth. Okay, during the life of a plant, the region that originally acted as a sink may become a source. Yeah, so sometimes it can change over because sugar stored in the leaf in the summer, acting as a sink, may be translocated to growing flower, bud, or stem in the following spring. So the bulb is now acting as a source. Yep, yep. Okay, so now we can take a look at the picture. Okay, so this is how the translocation happen. Okay, so at the start, the plant, the photosynthesis. Okay, so photosynthesis happen in the leaf. So here there will be lots and lots of sugar. Okay, so this sugar will go to the growing part of the plant. Okay, because it needs energy to grow. So here this will be the leaf will be the source. Okay, so where the sugar is produced, and then it will go to the sink where it end up. Okay, and then it can also go to the roots. Okay, so the root can also be the sink where the sugar can be stored here. Okay, yeah. So we recall the phloem is the outside part, okay, of the vascular bundle. Xylem is the inside part. Okay, yeah. So what is a sucrose? Okay, so normally the sucrose, the sugar is in the form of sucrose. Okay, what is a sucrose molecule? Is one side is glucose, one side is fructose. Okay, so that is a sucrose. It's the form that is suitable for transport purpose. Okay, so we also have amino acid that is transported. Okay, so in IGCSE we mostly focus on the sugar. Okay, so this is the structure of the phloem. It looks like this. Okay, so it also made of tubes. Okay, but it's different from xylem. Okay, so xylem is dead. Okay, it's a dead cell. It's just a tube. Okay, no cytoplasm. Okay, no nucleus. Okay, but the phloem is different. The phloem have cytoplasm. Okay, have nucleus. Okay, so now we have the tubes. Okay, and then we have the what we call companion cell. Okay, so this is the companion cell which has the nucleus and cytoplasm. Okay, so this companion cell is the one that will control what happened here. Okay, so they also have the seed plates. Okay, and then we have the gap in the cell wall that is joining the cells. Okay, and then we have the cytoplasm. Okay, so inside here we have cytoplasm. So the companion cell will help to control the movement. Okay, so inside the companion cell we have mitochondria, which help to produce the energy. Okay, so for the translocation, we need energy for it to happen. Okay, so we can see the arrow, it can go up and down. It depends on the situation. So depends like which part of the plant need the sugar, so it can go there. Okay, so yes. Okay, so normally the leaf is the source. Okay, so it produce. Okay, it produce the sugar. Okay, And then the flower is the sink because it needs sugar to grow. And then the stem is also the sink. Okay, it needs sugar to grow. Okay, the fruit, it can be sink or source. Okay, like example, during the winter time, okay, um, the plant, it needs energy, but it cannot have photosynthesis. So now it can also act as a source. So the seed is the same. Okay, it can act as both sink and source. Okay, the stem, okay, at the root is a sink. Okay, the tuber, okay, it can also act as source or sink because let's say during summertime the leaf produce a lot of sugar, then it gets stored in the tuber. Okay, it gets stored here, so now it is the sink. But during the winter, the plant, it cannot have the photosynthesis because there is not enough sunlight. Okay, so during the winter, the plants still need energy. So now the tuber can act as the source to supply the energy for the plants. Okay. Yeah. So that's the main point. Okay. So the source and the sink can change. Okay. So now basically we already finished all of the questions. I mean all of the notes. So now we'll look at the question. Okay. Study the diagram of root hair cell in figure 8.4. Which plant cell part is missing from this cell? Okay. So we will take a look at the root hair cell. Yep. Okay. Okay. Take a look at the root hair cell. Yeah. Okay. So which plant cell part is missing? What is missing? We know root hair cell, it has no chloroplast. Okay. No chloroplast because it is underground. It's not exposed to sunlight, so it does not need chloroplast. Okay. No photosynthesis. Okay, name the process which the cell absorb water. Okay, so how it can get water? It's by osmosis. Okay, so how it can get mineral ions? It is by um, diffusion. Okay, so it can happen diffusion and also active transport. Okay, so it depends. When outside, the mineral, okay, the mineral ion is very low. Inside is very high, then we need active transport. Okay. But if outside is high, inside is low, then it's just diffusion. Okay. So both way can happen. Okay. Next, study the experiment set up in figure 8.8. Complete the table to show the result and predict. You predict and give your reason. Okay. So just now the experiment. Yeah, this one. Okay. Let me just copy this. Okay. Yep. Okay, so what result we expect? Okay, okay, yep. So A is both side covered. Okay, so if both side covered, so all the stomata they will be covered. So we expect less transpiration. Okay, so the expected result, there will be no change, and the leaf remains healthy and not dry up. Okay, so how long we leave it for? Okay. Okay, it does not mention. So let's say we leave it for like uh 10 hours, something like that. Okay. So we expect no change because there is no water loss as all the stomata they have been covered or sealed by the petroleum jelly. Okay. So how about B? Okay. The upper epidermis is covered. Okay. So upper epidermis is covered. The leaf shrivel up. Okay, so the leaf dry up because most stomata are on the lower epidermis, so we have a lot of water loss. Okay, so normally lower epidermis will have more stomata. Okay, and then it has small amount of shriveling. Okay, so it's dry up a bit. Okay, because there are not many stomata on the upper epidermis. Okay, so we cover the lower epidermis. On the upper epidermis is only a few stomata. Okay, so it's protected from water loss by the waxy cuticle, the upper epidermis. Okay, so this one both side not covered. Okay, so this one we expect a lot of transpiration. Okay, so the water is very shriveled and dry up. Okay, so let me just find a picture to show. Uh, okay. Uh, yes. Okay. Okay, so this is what we call a, okay, shrivel up leaf. Okay, so it's dry up. Okay, because of the lack of water. Okay, so it shrivel up. So for the D, we expect maybe something like this happen. Okay, because both side not covered, so it can lose lots of water. Neither surface protected by petroleum jelly, so it has maximum water loss. Okay, next. Okay, so here we look at the lower surface of a typical dicotyledonous leaf. Okay, so this is a stomata. Okay, so this is the opening. Okay, so on figure, label part A, part B, and part C. Okay, so straightforward. Okay, so A will be stoma. Okay, B, this part here is the gut cell. Okay, C is the epidermal cell. Okay, epidermal cell. So the bottom layer of cell of the leaf. Okay, yep. Okay, next. Okay, the surface of the leaves of two species of plant was studied and the number of stomata per unit area was recorded. Okay, so they just take a leaf, okay, and then they count how many stomata, okay, on the leaf. And then next, what happened? Cobalt chloride paper changes color in the presence of water. So we have this cobalt chloride, okay, has paper, okay, which change color in the presence of water. Okay, pieces of cobalt chloride paper were attached to the upper and lower surfaces of leaf on both plants. The plants were set up for 1 hour during the day. Any color change were recorded. Experiment was repeated for 1 hour at night. The table shown below. Okay, so we have two plants. Okay, and then lower surface, upper surface. Okay, how many stomata? Okay, so for these plants, the um the stoma is mostly on the, so let's say this is the leaf. Okay, so for this cassia fistula, the stoma is mostly at the bottom, okay, of the leaf. So this one cassia fistula. Okay, this one Bahinia monandra. Okay, so this one, the, oh sorry, just now. Okay, okay, so this cassia fistula, the stoma is mostly on the upper surface. Okay, cassia fistula, and then this Bahinia monandra, the stoma, they are mostly at the bottom surface. Okay, Okay, so now we will attach the test strip at the top and then at the bottom. So we will attach the test strip. Okay, color change of cobalt chloride paper, which tell us water is present. Okay, so day and night. Okay. Um. Okay. So first we look at night. Night is easy, so we can see no water. Okay. So which is telling us no transpiration. Okay. No transpiration at night time. Why? Because at night stoma is closed. Yeah, okay. So stoma is closed. So this happened for most of the plant. So most of the plant at night time stoma is closed. So there is no transpiration. Okay, so that's why cross here. Okay. So during the day, you see the for this cassia fistula, they have the um stoma on the upper surface. So the upper surface we will detect water because stoma is there. Okay, for this Bahinia monandra, the stoma they are on the lower surface. So we will detect water at the lower surface. Okay, so that is the concept of this question. Okay, so question B, describe the differences in stomata distribution between the two species. Okay, so we discussed that. Okay, so for the C fistula, the stoma is mostly on the upper surface. For the B monandra, the stomata is mostly on the lower surface. Okay, so and then this is 22, this is 18. So which means in general, the monandra has more stomata than the C fistula. Okay, so that's the concept. Okay, next. Explain the color change to the cobalt chloride paper during the day. Okay, so what happened during the day? We discussed just now. Okay, during the day, the color change is due to water loss. Okay, water loss through the stomata because stomata is open during the day and then transpiration can happen. Okay. Number two, why no color change at night? Okay. So what we mentioned just now, why no color change? Because the gut cells and the stomata they are closed at night. Okay. So there is no transpiration or no water loss at night. Okay. So no water is lost, so we don't see any color change. Okay. Yes. Okay, question E. Uh okay, question D. Outline the mechanism by which water in the roots reaches the leaf. Okay. So how it happened? How the water move? Okay, so it happened in the xylem. Okay. So water enter xylem vessel through pits in the wall and then osmosis. Okay. Water move by osmosis. And then we have the transpiration pool that happen at the leaf. And then we have capillary action and cohesion of water molecules to help to pull the water up. And then we also have root pressure, whereby the root is having more water. Okay, so the transpiration can happen. Okay, question E. State and explain the effect of transpiration rate. Okay, when we increase humidity, what happened to transpiration? Okay, just now. Okay, so here. Okay, when we increase humidity, what happened? Yeah, okay. So we can see here when we are increasing humidity, transpiration is decreasing. Yeah, okay. The rate will decrease because there is a smaller gradient for the diffusion of water to happen. Okay. So how about when we increase temperature? Okay. when we increase temperature? This one. Yeah. Okay, so when we increase temperature, what happened? Okay, so increasing temperature, we expect transpiration to happen, but level off after some time. More energy or heat for evaporation. Water evaporate more quickly at higher temperature, and warm air can hold more water vapor than cold air. Okay, so these are some of the answers. Okay, so with this, we finish all the notes and the practice questions. Okay, for the unit 8. Okay, yep. Hold on. Okay. Okay, this is done. Okay, transport implant. Okay, so now we will move on to the workbook. Okay, yep, transport implants. Okay, moving on. Okay, so this one is very short, only 3 pages. Okay, okay, state 2 functions of plant roots. Okay, so what are the functions of plant roots? Okay, so they help to fix the plant to the soil, anchoring the plant to the soil so it does not fall down. Okay, absorption of water, absorption of mineral salts, and food storage organ. Okay, so the root of a plant can store food for winter and things like that. Okay, describe how root hair cell adapted for their function, making 2 key points. Okay, okay, how the root hair cells, okay, adapted to their function. Okay, yep, so this is the main point here. Okay, main features of the root hair cell. Okay, so they have a cell extension, okay, which increase cell surface area to make it more efficient for absorbing water and mineral ions. Okay, so main point, having high surface area. Okay, next. Okay, so this one, the diagram shows section through a root, list the parts through which water passes to get to the plant stem, putting them in correct order. Okay, so yep, it's like this. Okay, so it start at the root hair cell, the water go in, okay, and then it goes into the cortex, okay, and then it goes into the xylem, okay. So this is how water get into the plants. Okay, so here we have a section through a young plant. Label the position of xylem and phloem. Okay, straightforward. We know that. Okay, so xylem is inside. Okay, phloem outside. Okay, okay. State the function of xylem and phloem. Okay, this is straightforward. Okay, okay. Xylem transport water and mineral ions. Phloem transport sucrose and amino acids. Okay. Next, supplement. Describe how the structure of xylem vessel adapted to its function, making 3 key points. Okay. Yes. So what is the special structure of the xylem that make it very efficient for the transport purpose? Okay. So yes. Okay, let me copy. Okay. Okay, so structure of xylem vessels. Okay, so it is long thin vessels. They lack end walls. Okay, so no end walls. Okay, lack cell content, no cytoplasm, no nucleus. So it just got more space for the water and mineral ions to pass through. Walls may be lignified to provide strength and waterproofing. Okay, so it may be strengthened by lignin to make it stronger. Okay, next. Distinguish between transpiration and translocation. Okay, so transpiration is for water. Okay, translocation is for sugar and amino acids. Okay, making 2 key points each. Okay, transpiration is the loss of water vapor from the leaf through stomata by diffusion. Okay, translocation, movement of sucrose, sugar, and amino acid from sources to sink, from region of production or storage to region of use for respiration or growth. Okay. Two factors that lead to increase the rate of transpiration. Okay, we did mention that. Okay, high temperature and high wind speed will increase the rate of transpiration. Okay, next, multiple choice question. Okay, water is the correct order. What is the correct order of tissue through which water passes from soil to leaf? Okay. Okay, our answer is this. Yep. See, it starts at the root, it goes into the cortex, it goes into the xylem, and then the mesophyll. Okay, so we know that. Here. Okay. What we mentioned just now. So it starts at the root, and then it goes into the cortex, and then it goes into the xylem. Okay, so once it is in the xylem, okay, so once it's in the xylem, then it keeps going and then to the leaf, and then it goes into the mesophyll cells and then comes out from the stoma. Okay, next. Okay, the diagram shows an aphid feeding from phloem tubes. Okay, so if it is feeding the phloem tubes, okay, uh, what is the thing that it does not receive? Okay, so we know that, um, okay, so what is the thing that we expect it to receive? Okay, hold on. Oh, okay. Wait. Okay. What type of food it does not receive? Okay, so we know inside the phloem we have sugar, we have amino acids, okay, and then we have water, okay. So sugar, amino acids, water. So mineral ions not present. Okay. Yep. Okay. Next, describe the process of water loss from a leaf. Okay. So how the water is lost from a leaf. Okay. Okay. So basically, we have transpiration. Water enter the leaf through xylem. Okay. Yeah. Okay. Let me just copy this picture here. Okay. Hold on. Oh, okay. Okay. Let us take a look at this picture. Okay. Yes. Okay. So it starts at the root. Okay. It goes into the cortex and then go into the xylem, and then it keep going in the xylem, in the stem. Okay. Until it reaches the leaf. Okay. Reaches the leaf, and then it will come to the outside. It forms a thin layer on the mesophyll cells, and then it evaporate out. Okay. And then go out. Okay, so basically this is the process. Okay, yep. So water forms layer on the surface of mesophyll cells. Okay, what I mentioned forms layer. Okay, water evaporate into the air spaces. Okay, so now it will evaporate into the air spaces. Okay, into the spongy mesophyll, and then this create a high concentration of water molecule. So here we will have lots of water. Okay, so this is high water. So here this will be low water. So we have a concentration gradient. So the diffusion can happen. Okay, lower concentration in the air surrounding the leaf. So we have diffusion of water molecules through the stomata. Okay, okay, next. Yeah. A number of processes involved in the movement of substances into and around a plant. Complete the table by defining each term and explaining how it is important to a plant. Okay? Yep. So we'll look one by one. Okay, so first one, diffusion. Okay, diffusion. Okay, let me make it bigger. Okay, diffusion, definition, net movement of particle from region of higher concentration to a region of lower concentration. So basically, it's going down a concentration gradient as a result of their random movement. Okay. So how? Okay, it is important to a plant. Explaining how it's important to a plant. Explanation. Oxygen diffuses from air into a leaf. Okay. So uh gas exchange. Okay. And then another one, carbon dioxide diffuses in and out of a leaf. Okay. Gas exchange, and then mineral salt can diffuse from soil into root hairs. Okay, so three ways diffusion is important. Okay, so second one, osmosis. Okay, so what is the definition? Net movement of water molecules from higher water potential to lower water potential through partially permeable membrane. Okay, so this is the definition. Explanation. Okay, why osmosis is important? Is used to absorb water by the roots, and then the cell can absorb water by osmosis to become turgid, which keep the young stem rigid. Okay, so for support purpose, as we mentioned. Okay, so the osmosis help the cell to get water, so it remain turgid. Okay, so if there's lacking of water, wilting will happen. Okay, next, um, okay, active transport. Okay, so what is active transport? Definition: movement of particles through a cell membrane from region of lower concentration to higher concentration against concentration gradient using energy from respiration. Okay, so these are the key points. Okay, plants obtain mineral ions by active transport when the concentration of ion in the soil is lower than in the root. Okay, so when inside the soil is low, okay, then inside is high, so we need to, diffusion cannot happen, so we need active transport, okay, to move ion into the cell. Okay, next one, transpiration. Transpiration is the loss of water vapor from the leaf. Okay, so this is the definition and the explanation why it's important for the plant. Transpiration result in more water being drawn through the plant, carrying mineral ions from the roots. Okay, so it helps to bring water and mineral ions to um, every parts of the plants. Okay, next. Okay, yep. So here we have a diagram to show the um, section through part of a leaf. Okay, With reference to the feature label A to E, outline the process of water loss from the leaf. Okay. So now, okay, what we need to do, label A to E. A, B, C, D, E. Okay, and then how the water loss happen. Okay. Okay, so water will enter the leaf in the xylem vessels. Okay. So first, water will enter the leaf into the xylem vessels A, then passes in through the surrounding spongy mesophyll cells. Okay. So now water will go into the spongy mesophyll cells, which is B. Okay. A thin layer C form on the surface of the cell. Okay. So we form thin layer of water on the surface of the cell, okay, which evaporate and saturate the air into the air spaces. So now the water molecules will evaporate and stay in the air spaces, D, and then water molecules diffuse into the atmosphere through the stomata. Okay, so now finally water go into the atmosphere through the stomata, which is E. Okay, so that is the idea. Okay, explain why wilting occurs. So we discussed just now how the wilting occur due to the lack of water. Okay. Okay. When transpiration from the leaf loses more water than being taken in by the root. Okay. So the out is more than in. So now the cells in the young stem will be lack of water, become flaccid and stop pressing against each other. So yeah, so they become like this. So they do not press on each other. Okay. So they do not press on each other. Um, so they will lose their rigidity and they will wilt. Okay. So that's the concept. Okay, cool. This is done. Okay. So now we done all the workbook questions. Okay. And also all the textbook. Okay. So basically we finish everything for the transport implant. Okay. So this is also finished. Okay, so it is a short topic. We still have 30 minutes. Okay, so we will keep going. So transport in plant is done. Transport in animals. Okay, so we will get started on this transport in animals. Unit 9 of biology. Okay. Yep. Okay, so this one I also have sent to you already. Okay, so I think today. Okay, today I sent it. Okay, hold on. Okay. Okay, so transport in animals. Okay, so transport is also very important in animals. So we need to transport like oxygen, carbon dioxide, sugar, amino acids. Okay, all of these things we need a good transport system. Okay. Okay, so how the transport happen? We have the circulatory system. Okay, system of blood vessels with a pump and valves to ensure one-way flow of blood. Okay, so this is the idea of the blood circulatory system. Okay, so in IGCSE, we will learn two main types of the circulatory system: single circulatory system and double circulatory system. Okay, so single circulatory system we mostly find in fish, double circulatory system in mammals. Okay, so first we will look at the single circulation for the fish. We can see straight away the advantage. Okay, the advantage is simple. Yeah, okay. So it's quite simple, but the disadvantage. Okay, so what is the problem? Disadvantage, okay, is less efficient. Okay. Less efficient means we get less oxygen. Yeah. Okay. So less oxygen. Um, but it works well for the fish. Okay. So because fish is kind of smaller in size, so it does not need that much oxygen. So having a simple circulatory system is good enough. Okay. So now we'll look at how it works. Okay. So first we have the heart. Okay. Okay. So the heart is consists of one blood collecting chamber and one blood ejecting chamber. Okay. So we can see here. Um, this is the atrium. Okay, so we can see it gets blood from the um blood vessel. It collect, blood collecting chamber. So it collect blood. Okay, so this part, it is the ventricle. Okay, so ventricle is blood ejecting chamber. So the ventricle, it helps to pump the blood to the outside. Okay, so it sends blood to the gills where it is oxygenated. Okay, so this part is deoxygenated blood. Okay, so it is lack of oxygen because it's used by the cells in the body. Okay, so the oxygen is already taken by the body cells. Okay, so this deoxygenated blood. So now this blood is pumped to the gills. Okay, so the gills is exposed to the water so it can get oxygen. Yeah, okay. So it can get oxygen from the water. So now this part is oxygenated blood. Okay. So it then flow to all parts of the body. Yeah. So now it will flow to all the different parts of the body, supplying oxygen to the cells and then back to the heart. Okay. Now, the important point about single circulation, it goes through the heart once. Okay. One time for each complete circulation. Okay. So we can see one full cycle, it goes through the heart once only. Okay. For one complete circulation. Okay. So the problem with single circulation is pressure is lost. Okay. As the blood passes through the capillaries of the gills. Okay. So we want to have a good pressure. Okay. So we want to have a good pressure of blood, not too high, not too low, so that it can supply oxygen faster. Okay. But after the blood passed through the gills have many small capillaries. So the pressure will be low here and then here again, the pressure is low. So the oxygen is slow to get to the cells. Okay, so that's the problem. Not build back up until the blood returns to the heart. So it's inefficient. Okay, so the fish is getting less oxygen than the mammals. Okay, so that's the idea. Okay, but fish is a simple organism, so it does not need that much oxygen. Okay, so it works well for the fish. Okay, so how about double circulation? Okay, so double circulation, like for example, mammals, human, okay, will be having double circulation. Okay, so how it works? So we need to remember for the exam purpose, the circulation, how it happened. Okay, so remember for the heart is opposite. So this part will be right hand side. This part will be left hand side. Okay, it's always opposite. Okay, so the right side of the heart, it collect the oxygenated blood from the body. Okay, so in the body, it use the oxygen. Okay, all the body cells will use the oxygen. So this part will be deoxygenated blood. Okay, it goes back to the heart. Okay, in the atrium, and then it pumps to the ventricle, and then it goes to the lung. Okay, so here it is still deoxygenated blood. Okay, so in the lung, it will get oxygen. Yeah, it will get oxygen, and then carbon dioxide will go out. Okay, so now at this moment, we will get oxygenated blood. Yeah, okay. So now it will go back to the heart, okay, in the atrium, and then it go to the ventricle, and then it pump to the rest of the body. Yeah, okay. This oxygenated blood. Okay. So now the cycle is complete. Okay. Pump to the lung, and then the left side collect the oxygenated blood, and then it pumps to the body. Okay. So the double circulatory system, we can see one complete cycle, it goes through the heart twice. Okay. So it goes through the heart twice. Okay. So what is the good thing about this? It can maintain a blood pressure. Okay. So you see, going to the lung is only one organ. So we can have low pressure here. Okay. And then we coming back to the heart. Now we pump to the body. Now we pump to the body, we can have high pressure. Okay. So we can control the pressure. So it's more efficient. Okay. So for the low pressure situation, we can low pressure. To pump to the body, we need high pressure. So uh we will provide a higher pressure. Okay. So it's more efficient. Okay. Okay. Now we'll look at the heart. Okay. Yes. Okay. So mammalian heart. Okay. Example, human heart is made of muscle. So the muscle can pump the blood around the body. Okay. So the muscle is constantly active. Okay. So even when we are sleeping, our heart is still pumping. Okay. Yeah, our heart is still pumping. So so it's doing a lot of work. Okay. The heart is constantly working. So it needs its own blood supply to provide oxygen and glucose. Okay. So that's why we have this coronary artery. Okay. So the blood vessel that supply to the heart itself. Okay. What we call coronary artery provide oxygen and glucose. Okay. So now we have two types of blood vessels. We have veins and arteries. Okay. So veins is from body to heart. Okay. Carry blood to the heart is veins. Arteries carry blood away from the heart. Okay. So arteries go out of the heart, veins go in to the heart. Okay. So the heart has two sides separated by the septum. Okay. Yeah. Okay. So it has two sides. Okay. So this part will be the right. This will be the left. Okay. Separate by the septum. Okay. So this is the septum, the middle part. Yeah. Okay. So the middle part is the septum. So this is very important. Okay. Why? Because it separate the this one is deoxygenated blood and oxygenated blood. Okay, so this is very important. So we must separate the deoxygenated blood and oxygenated blood. So make sure we have high oxygen concentration to send to our body. Okay. The right side receive the oxygenated blood from the body and then pump to the lung for oxygenation. Okay. So as we mentioned before, so our atrium, this side will receive deoxygenated blood, okay, from the body, okay, and then pump to the ventricle, and then here it goes to the, okay, and then here it goes to the lung, okay. So it goes to the lung, and then it gets oxygen, okay, and then it's returned from the lung, okay. So now we get oxygenated blood, and then into the ventricle, and then so this one it goes to the body, okay. So that's how it happened, okay. Yeah, so now the left side will receive oxygenated blood from the lung, okay, at here, and then it pumps to the body, okay. Yes, so right chambers are on the left, okay, so it's opposite, okay. So right is here, left is here, okay. Next. Okay, four chambers. Yeah, okay. So right and left atrium. So the heart have four compartments. Okay, right and left atrium, they receive blood from the outside. Okay, right and left atrium. Okay, and then they will squeeze into the ventricle. Okay, squeeze into the ventricle. So ventricle here, two of them. Okay, and then and then the ventricle will squeeze into artery. Okay, okay, will squeeze here to the lung or to the body. Okay, and then surface view of the heart, we will see coronary arteries. Okay, on the surface of the ventricle. So here, if we look at the surface view, we will see the coronary artery. Okay, yeah. So we'll look at the parts that we need to know about. Okay, um, right atrium, left atrium. Okay, where blood comes in. Okay, right ventricle, left ventricle, where the blood goes out. Okay, and then these four blood vessels are very important. Okay, vena cava is the one that goes into here. Okay, pulmonary artery, it goes out. Okay, then pulmonary vein, it goes in from here. Aorta, it goes out from here. Okay, and then we also have muscular wall and septum, and then we have valve, four valve. Okay, semilunar valve at here. Okay, and atrioventricular valve. Okay, here. Okay, so valve, they kind of look like this. Okay, pulmonary valve. Okay, yeah, they kind of look like this, the shape. Okay, yeah. So they will open and close. So basically, it so this is closed. So when it open, water, the blood can go through. Okay, when it close, it stops the blood from going through. Okay, so the main function of the valve to stop the blood from backflow. Okay, so we don't want the blood to flow back. We just want the blood to go in one direction. Okay, so that's the function of the blood, the valve. Okay, next. So another key points, wall of the, okay, so let me just open again. Um, okay, 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 let me just copy this. Okay, Okay, so the wall of the left ventricle, so this one, left, this one is right. Okay, so left ventricle is thicker. Okay, thicker wall than the right ventricle because it needs enough pressure to pump the blood to all of the main organs. Okay, because it needs to pump to all the whole body. Okay, all the body parts. Okay, so that's why it's thicker. The right ventricle, it only goes to the lung. Okay, so this one is only going to the lung. Okay, so it does not need so much muscle. Okay, and then walls of atria much thinner than ventricles. Okay, so if we compare atrium and ventricle, the muscle is thinner. Okay, because the atrium, it only need to pump into the ventricle, so it just need to go to the ventricle. Okay, but the ventricles need to move the blood around the body. Okay, so that's a comparison. Okay, septum. Okay, this part here, septum, divide the heart into left and right. Okay, so this is quite important. Prevent mixing of oxygenated and deoxygenated blood. Okay, so we don't want the two type of blood to mix. Okay, okay, now uh how to control the blood flow in the heart. Okay, okay, so we can take a look. Okay, heart muscle in the atria will contract and build up sufficient pressure to move blood through the atrioventricular valve into the ventricles. Okay, so it will pump from atrium to ventricle. Okay, and then the valves will shut. The valve will close to prevent backflow of the blood. Okay, and then the muscles in the ventricles will contract. Okay, so now the ventricle will push the blood outside. Okay, of the heart. Okay, blood is forced through the semilunar valve. Okay, into the pulmonary artery and aorta. So once the pressure waves pass, semilunar valve close to prevent blood from the arteries from being sucked back into the ventricles. Okay, so again, this valve, it prevent the backflow of blood. Okay, so we just want the blood to go in one direction. Okay, okay, next, monitoring the activity of the heart. Okay, so the heart is very important. So we want to know our heart is healthy or not, our heart is normal or not. So we can check, okay, pulse rate. Okay, so like this, we put our two finger at our pulse, and then we just check in 60 seconds how many pulse. Okay, ripple of pressure that passes down the artery as a result of heartbeat can be felt as a pulse. The artery is near the surface of the body. Okay, like at our hand. Okay, and then another way is to hear the heart sound. Okay, this one, we hear the heart sound using the stethoscope. Okay, the stethoscope will amplify the sound of the heart valves opening and closing. Okay, next, but like normally without training, we don't know what to listen for. Okay, so we need training to know, to hear, um to identify when there is a problem. Okay, so training is needed. Okay, next, ECG, electrocardiogram. Okay, so cardiogram is kind of like this. Okay, so we will put the electrode attached to the machine and stuck onto the surface of the skin at the arms, legs, and chest. And then we will see electrical activity of the heartbeat, okay, at the computer. Okay, so at the computer, we will see this signal here. Again, like normally we don't know how to interpret this graph. Okay, so we don't really know what is going on, but with training, like doctor or nurse, they know how to read this graph. Okay, okay, next, Effect of physical activity on heart rate. Okay, so we know that if we have physical activity, if we exercise for a while, our heart rate will increase. Okay, we know that. Heartbeat is a contraction that forces blood into the lung. Okay, and the body. So heartbeat, the normal heartbeat is about 70 times a minute. Okay, more if you are younger, and then the rate will be lower if you are fit. Okay. Okay, so we can check the pulse at the wrist or the neck. Okay, like what we're doing here. Okay, so during exercise, the heart rate will increase from the resting state. Okay, yeah. So let's say this is our resting state. When we start to exercise, our heart rate will increase. Okay, and then it stay high until physical activity slow down or stop. And then after exercise, the heart rate will return to normal. Okay. After exercise, it will slowly return to normal. Okay. Um, yes. Okay, so we'll see the blue color and black color. Okay. So the blue color is untrained person, black color is trained person. Okay. So untrained means this person they do not exercise normally. This one, the black one, they exercise regularly. Okay, so their pulse rate is lower in general. Okay, so the black color is lower than the blue color. Why? Because they normally exercise, they have a strong heart. Okay, and then they have a large, okay, lung capacity. Okay, so they don't need to beat so many times to get the same amount of oxygen. But for a person that is lack of training, their heart is not strong, so they need to beat more times to get the oxygen that they need. Okay. Okay. During exercise, the heart rate will increase to supply the muscle with more oxygen and glucose. So these are needed to provide energy for the muscles contraction. Okay. So they have enough energy for muscle contraction. Okay. Next. Yeah. Okay. So our heart is very important. If we don't take care of it, we will get a heart disease. Yeah. Okay. So the most common type of heart disease is this one. Okay. Blockage of coronary arteries. Okay. So we can see here. Yeah. Okay. So the normal artery is like this. The blocked artery is like this. Okay. So it's the plaque. Okay. So we got something like built up in the blood vessel. So the blood now is very difficult to pass through. Okay. So if that happen, okay, we might get a heart attack. Okay. Yeah. So the coronary arteries, if they are blocked, they cannot supply the heart with oxygen and glucose. So the muscle will stop contracting and die. Okay. So we'll get a heart attack. Okay. Risk factors for coronary heart disease. Okay. So what are the things that will make it easier to get a heart disease? Okay. So we can take a look. Okay. Poor diet, too much saturated animal fat, okay, will cause these fatty deposits, okay, this fatty deposit on the blood vessels, okay, which will block the blood vessel and cause the blood clot, okay. Another problem, obesity, okay. So for people who are overweight, the heart need to work harder, extra strain on the heart, okay. And then for people who are overweight, it's also more difficult to exercise, okay. Smoking. So smoking is bad for the heart because nicotine will damage the heart and blood vessels. Okay, stress. Stress will increase the blood pressure, okay, which can result fatty material collecting in the arteries, okay. So stress is also not good for our heart. Lack of exercise, okay. So if we do not exercise regularly, the heart muscle will loses the tone, will lose the muscle, which means it's less efficient in pumping blood. Okay, so we cannot get enough oxygen. Genetic predisposition. Okay, so sometimes it's because of the genes that we get from our parents, passed from one generation to the next. They have more risk, okay, for the heart disease. Age, in general, the risk will increase with age. And sex, okay. Normally, males are more at risk than females to get the heart disease. Okay. Okay. Okay, final parts. Okay, how we prevent the heart disease? What can we do to prevent heart disease? Healthy, balanced diet will lower the chance of obesity. And then we need to lower the intake of saturated fat to reduce the build up of fatty deposit and blood clots. Okay, so we want to prevent this thing from happening. Okay, regular vigorous exercise is important to increase our muscle tone so we get more muscle, especially our cardiac muscle, our heart muscle. Okay, so now can improve blood flow from the heart. So we don't need so much effort to keep pumping. Okay, next sample question. Okay, name the two blood vessel A, B. Okay, so what's A? A is vena cava. Okay, so B is pulmonary uh, artery. Okay, if I remember correctly. Yes. Okay, A is vena cava, B is pulmonary artery. Okay, so C is aorta, D is pulmonary vein. Okay, which of the blood vessel carry oxygenated blood? Okay, so this one is the oxygenated blood coming from the body, okay, going into the ventricle and then going into the lung. So here is still deoxygenated. Okay, so once it's back from the lung, so now it's oxygenated and then oxygenated, oxygenated. So our answer C and D. Okay, oxygenated blood. Okay, name structure E and state the function. Okay, so E, this one is a valve. Okay, which stop the blood from going backwards. It stop the backflow of the blood. Okay. Okay, this is good. So now we finish up to here. So next section is talking about the blood vessels. Okay, so the different kind of blood vessels, their function, okay, things like that. So I think next time we can finish this part. Okay, so we are running out of time. Next time we can finish. Okay, so now our progress is at the transport in plants. Okay, so after today's class, you will take a break. Okay, today is 12. You will take a break until the next class is on Sunday, okay, 24th of August, 8:30 to 10:30 a.m. Okay. Yeah, so if you want to make it earlier, you can let me know. Yeah, otherwise we will keep it like that. And then after that will be every Sunday. Okay. So will be every Sunday. We will be having the class 2 hours in the morning. Okay. So this is the current planning. Okay. So if any changes, you just let me know. So otherwise we just continue like this. Okay. So yeah. So basically we will stop here. So next time we will continue. If no other question, we will stop here. Okay. See you on 24, the next Sunday. All right. Okay. Thank you. Thank you. Bye bye.